Okay. Hi, this is Maggie Lewis. I'm a PhD student in the Department of Entomology at the University of Maryland, where I study the biology and management of spotted-winged Drosophila, an invasive vinegar fly that has recently emerged as a primary pest of soft-skinned fruit, particularly fall-bearing raspberries and blackberries. In caneberries, we often see that periods of peak SWD activity overlap with two primary raspberry pathogens, Botrytis and Cladosporium fruit rot. In previous surveys of raspberry fruit, we found that larval SWD co-occur with and feed on both Botrytis and Cladosporium. They overlap relatively infrequently, but this does suggest that there may be an association between these different classes of pests. Apart from this, however, relatively little is known about their interactions, but they may impact insect and pathogen management within raspberries. Understanding these interactions could have important implications for management. For example, if the fungal associations that we observe during the larval life stage extend to the adults, it's possible that adult flies could act as a vector for fruit rot pathogens, transmitting these diseases to healthy raspberry fruit. Over the course of the past couple of years, our lab has conducted a series of field and laboratory-based studies aimed at better understanding these interactions. One aspect of this work includes a survey of adult fungal associations. In these surveys, we went out to raspberry and blackberry fields, hand-collected adult flies, brought them back to our lab, and isolated fungi from both the exterior of their body as well as from within their gut. By taking these two sets of measurements, we were able to both quantify the fungal community that adult SWD associate with, and also look at the mechanism by which these interactions are potentially occurring. Are the adult flies landing on a moldy berry and picking up spores on their legs or some other part of their body? Or are they actually taking the time to stop and feed on the fruit tissue? In 2018, we sampled a total of 34 flies and isolated 22 different genera of fungi from their bodies, including both Botrytis and Cladosporium, as well as 6 other genera of fungi that contain known plant pathogens. In the table on your screen, you can see that for each of our 8 potentially pathogenic isolates, we have the total number of flies surveyed out of 34 that were carrying fungal propagules on either the exterior of their body or within their digestive tract, indicating feeding. Overall, the majority of fungal isolates did come from the exterior fly body, suggesting that they're picking up these spores when they land on infected fruit tissue, but it's also possible that if they feed on the fungus, the spores break down as they pass through the digestive tract. Overall, these surveys do indicate that adult flies are associating with a fairly diverse fungal community, and we also see that these flies are capable of carrying fungal spores on their body, which introduces the possibility of a vectoring relationship. To better understand the potential for this vectoring association, we've also conducted laboratory-based vectoring assays. These assays were conducted using no-choice, worst-case scenario laboratory conditions, and were really designed to be a proof of concept study that could justify future lab-based, more field-realistic work. In these assays, we took adult spotted-winged Drosophila, held them on a sporulating Botrytis or Cladosporium culture for 5 hours. At the end of that period, we removed the flies and transferred them onto individual sterile agar plates, where we held them for 24 hours. At the end of that 24-hour period, we removed the flies and transferred them to a second sterile agar plate, and this process was repeated 2 more times for a total of 4 vectoring time points, ranging from 0 to 72 hours after exposure. So once we removed the flies from the agar plates, we held onto them and monitored them for fungal growth. If fungal growth was present, it would indicate that the flies were capable of transmitting these pathogens, at least under these sterile laboratory conditions. In this table, you can see the percentage of flies at each of our vectoring time points that were able to spread either Botrytis or Cladosporium to the sterile agar. And you can see that for both pathogens, we had nearly 100% vectoring through the 72-hour time point. What this suggests is that once these flies pick up the fungal spores on their body, they are able to transmit these pathogens, and these vectoring relationships are potentially persisting for a prolonged period of time. Of course, we will need to conduct future, more field-relevant studies to understand how well these laboratory results translate to a field-realistic scenario, and there are a number of other aspects of these interactions that we need to understand as well. For example, how do these pathogens impact spotted-winged Drosophila infestation patterns, or does larval feeding impact fungal growth? But ultimately, by understanding these and other aspects of SWD's interactions, we may be able to advance raspberry pest management, developing integrated management programs that account for both insect and pathogen pests. If you would like to learn more about these and other spotted wing Drosophila research that's currently going on in the Hamby Lab, please feel free to visit our website, hambylab.weebly.com, and you can also find my contact information there if you would like to learn more about this project. Thank you.